Happy day to everyone. I Suganya from Department of Pharmaceutical Technology. Today we are going to discuss on the topic media for industrial fermentation in bioprocess engineering. And the topic is mainly based on the production of the bioproducts and the pharmaceutical products in which microorganisms plays a vital role. The microorganism requires water, energy sources like carbon, nitrogen, minerals and possible vitamins for the growth. Especially oxygen is required if it is an aerobic process, and carbon dioxide is required in certain anaerobic process also. So here we are going to now today see the criteria for the media preparation. There are 7 general good criteria which has been followed in the preparation of media. The first one is a maximum yield of the product or the biomass which is required, and the second one is production of the maximum concentration of the product which is being done with the maximum rate of production of the formation, and the other one is minimum yield of the undesired products which has to be removed in the further downstream processing, and then the consistent quality of the available of the raw materials throughout the year, and there should be minimal problems during the media making and the sterilization process. And there is other important aspect to be considered in every media preparation is the media which is being prepared should not disturb the process of aeration, agitation, extraction, purification, and also it should be very comfortable in the waste treatment as well. And then when we see about the media preparation, there is a basic formula which has been followed in all the aerobic fermentation process. The first is what is the substrate we are giving and what are the products which have been derived. The substrate is mostly carbon and the energy sources along with it nitrogen, oxygen and other requirements which gives us as a biomass and the product along with the generation of heat, carbon dioxide and water. And we know the carbon is a main substrate which plays an important of dual role. One is in the process of biosynthesis and in the process of energy generation. So at the end of the process we are going to check what is the yield of the cellular coefficient, and the yield is calculated with the amount of dry matter produced in the process with respect to the carbon which is being utilized. And since it is an aerobic process, the oxygen demand should be calculated before we starting the industrial fermentation process, and that is been depends on the design of the media. And now we are going to see the main important factors which is being used in all the industrial fermentation is water. Here the water is being not only required during the process of production of the media, it is also required in certain accessory services like heating, cooling, cleaning and also the rinsing the biofermenter. And here in the production of process of certain byproducts like brewing industry and mashing industry, the mineral contents which is present in the water is also to be considered as an important factor. And in certain other process like in the beer production or in the wine production, we can treat the deionized water to which we can add the salts and adjust the pH during the production process. And one of the important factors in every industry is followed is how efficient we can reuse the water for the other process. Like when it is being used for heating purpose, that is also used in the heat generation, and the same water can be used in the cooling purpose also. The reuse of the water is being considered as a high priority in all the industries. And now we are going to see about the energy sources. When we see it is an energy sources in the media components, most of the chemoorganotropes like the organisms which has been using the carbon as a sole source, and certain lipids and proteins also there. There are microorganisms which can use hydrocarbons or even methanol as a carbon source for the energy production. Now we are going to see detailed stuff about the carbon source. When we see about the carbon source, first important carbon source is used as the carbohydrates. We will see in the next slide. And here there are main factors influencing the carbon source, or what is the main product which has been produced in the fermentation at the end of the fermentation. And in certain pharmaceutical ingredients, when we are preparing the pharma products, it is very important to see what is the purity of the carbon which is being used as a choice of the substrate. And the next important stuff is that carbon source which has been used should not change its activity in the process of sterilization, because sterilization process is one of the factors which is being done prior to the inoculum of the organisms. And then there are certain influences of the government legislation and local laws to be followed when we are using that carbon source, which is a waste product of the different industries. And the commonly used carbon sources are carbohydrates, hydrogen, hydrocarbons and its derivative. And when we see the next slide about the carbohydrates, there are different variety of sources available. For example, we can use as a maize and grains, potatoes and cassava as a sole source for the starch, and we can use sugar cane, sugar beet as a sole source for sucrose, and in the brewing industries malt has been used as a sole carbon source, and there are other certain compounds which can be used as a carbohydrate source in the media preparation. The next we are going to see about the hydrocarbons, which is cheaply available in the production process. And certain, we know what is alkanes. Alkanes is nothing but one of the hydrocarbons which has hydrogen and carbon as a basic ingredient in it. And there are certain compounds which can be produced with using hydrocarbons alone, like organic acids, amino acids, vitamins, cofactors, antibiotics, enzymes and proteins. In the next we are going to see what are the general nitrogen sources which is being used. We know microorganisms requires carbon and nitrogen as a basic energy sources. When we see in the nitrogen, inorganic nitrogen which is being supplied as ammonia gas and ammonium salts or in the form of nitrates, and there are nitrates which is being commonly available as alkaline drift in the metabolic process. And organic nitrogen may be supplied as amino acids and which can be involved in the metabolizing process. In the large scale industries, the carbon source will be as a byproduct of some other industries. Similarly, nitrogen source can be also a byproduct of certain industries where corn steep liquor, soy meal or yeast extract, which is a byproduct of another industries, can be used as a sole nitrogen source. And in next we are going to see about what are the different factors influencing the nitrogen source. One is, the main factor is control mechanism. When we see it as a control mechanism, there are certain enzymes be added to convert the sole carbon nitrogen source into ammonia for the further use in the production process. And then in antibiotic production, many microorganisms influenced by the type of the nitrogen source which is being present in the culture media. So the selection of the nitrogen source is also one important factor to be considered while the media optimization process. Now we are going to see about the different types of carbon sources and different types of nitrogen sources which is available and which is being used already in the industrial fermentation process. The first cheap and the byproduct of the sugar industries and mostly having the most of the trace elements also present in the source is the molasses, and followed by molasses, whey from the bioproducts of the dairy is being used in the large quantity, and others are the malt extract, hydromolasses, sulfate waste liquor or the sole source of carbon. And in nitrogen sources, it has been used as inorganic salts like ammonium salts, ammonia, urea and soy meal peptones, along with corn steep liquor which is being produced in the starch production from the corn, is being the different types of sources. And now we are going to see about the other substances which is being added in the media while industrial fermentation, that is oils and fat. There are vegetable oils or fatty acids or linoleic acid which is being used in the industries, mainly for oils and fat was mainly used as an anti-foaming properties. We know in the aerobic fermentation process, oxygen demand is an important factor to be considered. In that oxygen demand, when we supply oxygen from the bottom of the fermenter, it will create foams and which will block the surface transfer of the air to the medium. So anti-foaming agents like oils is being used to avoid the foam production in the fermentation process, and methylolate is a sole carbon source in the cephalosporin production which is also act as a anti-foaming agents by itself. Now we are going to see about the minerals which has been added as a requirement for certain microorganisms. There are different minerals which has been used, and there are certain minerals which have been already present in the products like molasses, corn strip, liquids. Yet it is to be considered as an important factor because certain microorganisms will not grow if there is no minerals present in the medium. So examples for the minerals, we know magnesium, phosphorus, potassium, sulfur, calcium, chlorine, along with the certain micro minerals like cobalt, copper, iron, manganese, molybdenum, zinc. In the synthetic media, the minor elements can be added deliberately while we prepare ourselves, but when we using the raw material from the other industries byproducts, there are certain minerals which is already present. Without knowing the level of that, we can't add the extra minerals to it. Next, we are going to see about the growth factors where the microorganisms fully synthesize and grow in a better manner. The growth factors are added mainly to start the process of the growth of the microorganisms. We know when we add a microorganisms to a sterilized medium, it is very important role of the growth factors and the carbon and nitrogen source to utilize by the microorganisms and to produce the biomass of it. In the media formulations, the growth factors like vitamins, amino acids, fatty acids and certain sterile compounds are required for the effective growth of the organisms. And then now we are going to see about the other metabolic regulators which is being used in the media. When we see metabolic regulators, it is not just the microorganisms growth and it is being using for their own primary compounds. It is also important to add additives like precursors, inhibitors, inducers depending on the type of the product which is being produced, because since it is very easy in the production of certain organic acids, but when we come to the drug production in the pharmaceutical industries, it is very important to check whether the microorganism is producing the desired product instead of producing any other extra undesired products. Here the precursors which has been added to initiate the process and inhibitors has been used to avoid the undesired product production, and inducers are used to increase the rate of production of the fermentation process. And then the oxygen, as we talking from the beginning in the aerobic process, the amount of oxygen demands in the fermentation process is mainly depends upon the organisms which will be used in the production process, and there are certain availability of oxygen is being influenced by the fast metabolism of the organisms, and rheology is nothing but the viscosity of the medium which is being present and how well that medium helps in the production process. And to avoid the foams in the supply of the oxygen, we also add antifoams which we already discussed in the oils and fat stuff. And then we are going to see about what are the different components available in the sole industrial fermentation media. The first, when we see about the molasses, the major carbon source is sucrose, where it has very limited supplements of nitrogen, and it also has an abundant level of phosphorus and sulfurs, but least level of potassium and calcium in it. And vitamins, most of the vitamins is already present in it, where biotin may be the limiting factors, and the minor compounds like betaine, organic acids, wax, pigments and silica is already present in the molasses, which is the cheapest source and the byproduct of the sugar industry. Similarly, in the malt wort, the glucose is the major carbon source, amino acids has been present as a nitrogen source, and most of the minerals are present where the zinc is the limiting factor, and vitamins are already present, and the minor compounds which is present in the malt wort is maltodextrins. When we come to the grape must, glucose is limiting, it's in the small amount where the fructose is a major carbon source which is present in the grape must, and it already contains peptones as a minor products, and it has a wide range of vitamins. We know the grape is a fruit where we can get a large vitamins, and it also contains a mineral sentence. Next is cheese whey, where in the cheese whey it is a byproduct of the dairy industries, and lactose is a major carbon source. And all we are talking about the different energies like carbon, nitrogen, oxygen supply, minerals, vitamins, and before starting any fermentation process, the major factors which has been already to be completed is media optimization, like what is the level of carbon to be added, what is the level of nitrogen, what type of carbon to be added and what type of nitrogen to be added, what are the mineral factors, what is a gram to be added in it, everything to be optimized in the media. And that optimization is a very huge chapters to be discussed, and that optimization, the optimized media will help us to produce the desired product in the short time. And there are different types of optimization where the Plackett-Berman method and response surface methodology is being used. Now we are going to see about the possible questions in this topic. One is the important criteria for preparation of good media. The next is different types of metabolic additives has been added in the media preparation, and the next is list of carbon sources used in the industry level, and the other important question is what are the factors influence the choice of the carbon. Thank you so much.